Sweet.
Jackson, great debate. Tons of fun. We had like 100. I think we had about 320 people at one point. Human
Yep,
evolution,
so
man. That's one of my favorite topics for sure. Jackson and I
yeah.
debated that
Mhm.
several months ago, actually. Human evolution.
Yeah, I couldn't pick out a winner from that. I think we both kind of countered each other pretty well.
I enjoyed it. I enjoyed that
Yeah.
debate. I think we got deep into a lot of the so-called hominids and transitionals. But that's my question to you, Jackson, is how do you feel about, and you may even disagree with this, actually. That's why I don't mind pushback. But I find after examining all the best so-called examples of ape to man evolution, so-called basal forms of humans or primitive forms, as they'll call it. Right? They'll pick up the most anomalous bone in the fossil record, and they'll say, see, this is basal because there's some anomalies when you compare it to extant humans. But from my investigation into this, I've been able to, I believe, sufficiently categorize every so-called transition or example of a basal human as either fully human, degenerated human, fully ape, degenerated ape, or amazingly an artificial species. Like I've got a couple articles in front of me, including a couple papers that talk about how Australopithecus sediba and Homo habilis may indeed be the result of the accidental mixture of ape and human bones, right? So that's what I mean by artificial species, is in that which is called Australopithecus sediba isn't actually a real species. It's a mixture. What are your thoughts on that, Jackson?
Well, there are two good specimens of Australopithecus sediba, so I think it's pretty unlikely that they're an artificial species. And habilis might be, and Rudolfensis might be the same species, or they might be an Australopithecus. It really doesn't matter what you call it at that point. But yeah.
Actually, John, if you could share my screen. Can you see my screen, guys?
No, born 100 years late. I only have like 3.
John,
There's
is
only
my
3 of them.
Oh,
Yeah,
you can see my.
Yeah,
Okay.
your screen.
Okay.
But the thing about sediba is they're not really thought to be our direct ancestor. They're more of an offshoot.
Right.
They're the timeline and some of the features doesn't really match with what we would expect from a true human ancestor.
Well, from my experience debating, I'm hearing a little bit of feedback. From my experience debating some pretty militant evolutionists, they'll look to sediba and habilis. For example, Gutsick Gibbon, she's got a video titled something similar to, the perfect transitionals, something like that, or a creationist worst nightmare. And the two she picked out are Australopithecus sediba and Homo habilis. But what's funny is,
I
those
wouldn't have picked sediba.
Yeah. And those are the two most disputed. Like, for example, even if it wasn't an artificial species, as you pointed out, just in 2018, 2019, there's many paleoanthropologists pointing out that Australopithecus sediba has been ousted as a direct ancestor to the Homo genus, right?
It's more.
But then there's also those right here that I was pointing out. Just to demonstrate what I was saying is true. Human missing link species may be jumble of species. And Ella Bin and Yoel Rack, they've pointed out that Australopithecus sediba is a mixture of Homo and also apes. And also, and last thing I'll say too, and I'll get your thoughts, Jackson, famous paleoanthropologist Lee Berger, he's actually recently admitted that Australopithecus afarensis, right, Lucy, the famous Lucy species, is a chimeric species. It's the mixture of human and ape bones as well. So Lucy being one of the most favorite so-called examples of human evolution over the years is now even being looked at as a possible chimeric species. So my point is, it doesn't really seem like there's any really strong examples, when if human evolution is true, I feel like there should just be so many examples that it's just undeniable, it's overwhelming, and I just don't think that we really see that. Yeah, go ahead. What are your thoughts, Jackson?
Well, there are a couple other specimens of Afarensis and Lucy, like the big man and the Salem child, that are partially complete skeletons, and there are like hundreds of other incomplete just fragments. So I don't think it's a chimeric species, but there's one almost fully complete specimen of Australopithecus prometheus, or which might be an early Africanus, that shows that it was trapped in limestone, Sturgeon cave. So we know that all the bones belong to the same individual conclusively. And it's not on our direct lineage, most likely, but it's an offshoot showing the progression.
Right. But that seems to be my biggest problem, is the best so-called examples of a true transition from ape to man, Australopithecine-like ancestors up to Homo habilis, up to Homo erectus, and eventually Homo sapiens. They always consist of such a scattered bone collection, right? Lucy here was originally found in hundreds of pieces, and that's how you can even get chimeric species in the first place, is because, like, for example, when you hear, because you keep saying, they got this many specimens, and when you hear defenders of evolution say, we've got 400 specimens of Lucy, people think, oh wow, they got 400 full complete skeletons. Right. So, for example, this is an example of what they mean by 400 specimens. It's 400 fragments here. Everybody on the panel can see. So they turn this into that. I mean, it's amazing. From these fragments, they know what Lucy looked like and what Lucy's husband and child looked like.
a lot of assumptions went into putting that together, I think, just like the geologic column theory. Don't get me wrong, it's not bad to try to guess things, but whenever you put things together just the way that you think they fit, and they just happen to almost fit that way, it doesn't really prove anything. It's a theory, don't get me wrong, but it hasn't been observed. I mean, it hasn't gone through a scientific.
I wouldn't even call it a good theory.
It's a hypothesis at best.
Well, they'll say it's a theory, but something way, way before a theory is a hypothesis.
Yeah, I mean, a theory at least has to be tested once, correct? I mean, you kind of hypothesize about it, then you test, observe, repeat, and form a theory as you go, kind of thing.
For example, if I had a theory, I had an idea that a cow, we know cows can jump. So I mean, if you feed it enough, train it enough, eventually it'll be able to jump over the moon. we know there's no evidence for that. There's nothing supporting it. So it's always just going to stay as a hypothesis, and that's exactly what we see with human evolution. They have an idea, but it's not supported by the data, which means it's just going to remain a poor hypothesis. And I'd be curious as to Jackson's thoughts here too, because they'll try and say, well, you've got your Australopithecines, 6 to 8 million years ago, and then eventually you got Homo habilis, which they'll say is a perfect transition of Australopithecus-like ancestors to the Homo genus. But what we actually see is extensive coexistence of even the earliest Australopithecines and man. And I have a list of examples as to how we know they coexisted. And we find human limb bones, human jaw bones, human hands, foot bones. We find human footprints, human bones, shelters, stone tools, butchered bones, showing that it's humans, obviously the ones doing this butchering, and also humans being buried with Australopithecines. And last thing I'll say, examples of humans eating robust Australopithecines. So my point is, we see such overlap, which is actually what we would expect after the flood and post-Babel or even pre-Babel, is we would see a lot of this coexistence, right? Yeah, what are your thoughts on that significant coexistence we see, Jackson?
Well, nothing I've read has suggested that humans in their modern forms survived alongside Australopithecines, but maybe Homo habilis or some other early Homo species did. And I know Australopithecus anamensis and Australopithecus afarensis inhabited the same area at the same time, but it's inferred that anamensis may be its ancestor. But maybe one population diverged and became afarensis, and one population kind of remained fairly similar in anamensis. Might be one explanation for that, but I'm not familiar with humans living alongside Australopithecines.
Yeah, you find example after example of it. For example, here's a quote from famous paleoanthropologist Richard Leakey. I see no reason that bands of Homo would not have killed and eaten robust Australopithecines when they could, just as they killed and ate antelopes and other prey animals. There's also a quote from
I know what you're talking about now. The robust Australopithecines, like Paranthropus, they did live alongside like Homo habilis.
Right. But remember, if people like even Lee Berger are admitting that, well, Australopithecus afarensis may be a chimeric species, mixture of Homo and ape bones. Well, that means by definition, humans must have lived alongside or coexisted with the Australopithecine. Now, I know what you're saying is, well, maybe it's a basal form of humans, right? You're saying, well, maybe it's erectus, but that's the point. Is the data that we find pertaining to so-called early humans. I'm trying to pull up the paper here on Homo erectus. I can't find the paper, but I do have an example here that Homo erectus has evidence for purposeful navigation, has evidence for being able to speak, but Homo erectus also has evidence for what's called reductive evolution. And that's where you have these subpopulations isolating themselves. And what results is inbreeding and accelerated genetic degeneration. So they'll pull up these anomalous features, these anomalous bones, and they'll say, see, this is basal to extant humans today. When in fact, no, what we're looking at is something that's fully human, but it's riddled with pathology, it's riddled with disease, it's riddled with degeneration. So where they're saying is, this is basal, this is primitive, it's actually just an example of a fully human people group experiencing reductive evolution, which isn't evidence for obviously ape to man evolution. Yeah, go ahead, Jackson.
Well, we don't have DNA for erectus, but we do for Neanderthals, which aren't our ancestors, they're more our cousins. But we differ by 202 base pairs on the mitochondrial DNA, which indicates there are different species. So I don't think there's a degenerated human or a different human.
If my screen's still shared, you can see the drastic difference in understanding pertaining to Neanderthalensis. 1856, they articulated and represented him as this brutish half ape, half man. But today, for example, in 2015, the reconstructions have drastically changed. And this is based on the fact that now we know that Neanderthals buried their dead, made musical instruments, tools, cosmetics, jewelry, and purses. Their DNA shows that they were human and interbred with us. I mean, we literally have their genetics. And the reason why they are different in terms of their genetics, as in there's a slight divergence, right? Even though they're still 99.7% similar to us, right? Every human today is 99.999% similar. Neanderthals, which are an extinct people group, they're 99.7% similar. Well, we'd expect their genetics to be slightly divergent because they were the most inbred people group on the planet, actually. And they accumulated a lot of recessive mutations came to the forefront because they existed in small tribes. Therefore, they were highly inbred. And what would result is rapid fixation of new genetic variants. So that explains why they're slightly different in their genetics. But we actually have paleoanthropologists that will admit Homo neanderthalensis should be considered a subspecies of Homo sapiens rather than a separate species. I think the only people that want to say Neanderthals are a sister species or a separate species are those that are so committed to the ape to man evolution story that they'll say, well, sure, according to the modern definition of species, then yes, they would be the same species. But, assumption after assumption pertaining to human evolution, they can't actually be a descendant of Homo sapiens, so therefore they'll put them as a sister species. But the actual genetic data and also the fossil data would just suggest that they're a variant of human, but they are degenerated due to inbreeding. Yeah, go ahead, Jackson.
Well, if they were degenerated due to inbreeding, and all humans started out with 2 individuals, why aren't we all the same amount of inbred?
That's a good question, actually. That would be because, and I think you and I discussed this in our debate as to what the fundamental difference is between our model and your model when it comes to the origin of genetic diversity. For example, how do you explain, from an evolutionary starting point, the origin of most or all genetic variation?
Well, I don't know. I'm honestly don't have a good explanation for it. I know other people do. I'm just not the best representative for some of this subject matter.
No, that's fine. That's fine. I'm not referring to like the origin of the first life or the first cell. I'm talking about the DNA variety, the DNA differences that exist within species and across species. I'm just talking about how I'm really hearing some feedback. So I don't know. Hopefully whoever's not talking, just mute. I don't think it's you though, Jackson. So for example, here, if my screen's still shared, it says the ultimate source of all genetic variation is mutation, right? The evolutionary model, they'll say that mutations add genetic diversity, right? Because mutations are adding something that was not previously there. So the evolutionary model looks to mutations as being the source for all genetic variation. And eventually you have processes like recombination, which can scramble up that genetic variation, those DNA differences, creating new varieties and new species. But our model, as a creationist, we look to created nuclear heterozygosity. So what that, it sounds fancy, but all it means is the original created types or kinds, they would have been front loaded with genetic variation. So you look to mutations as being the ultimate source. We actually look to most genetic variation as being not by mutation, but by being the result of design, initial front loading at creation. So that means Adam and Eve would have had no mutations. They would have had no genetic mistakes, no genetic defects at creation. And therefore, there wouldn't have been an inbreeding problem. Yes, there would have had to have been inbreeding the first couple generations, but there'd be no mutations those first few generations to actually come to the forefront and lead to genetic damage. But the evolutionary model, there's a problem for you, Jackson, because you explain genetic variation as being the result of mutation. So yeah, you actually have an inbreeding problem when it comes to the out of Africa population bottleneck, for example, because you have all of these mutations in hidden recessive forms that are going to come to the forefront and be manifested, leading to accelerated genetic damage. So that's how we don't have a mutation problem or an inbreeding problem, just to answer your question. Yeah, go ahead, Jackson.
Well, even if Adam and Eve had a perfect genome, their offspring wouldn't. They would be damaged by the sun's rays, or genetic code would be damaged to some extent somehow. But even if it wasn't, you have long after the fall, you have Noah's Ark, which is another severe genetic bottleneck where there are only 3 pairs. So we're back to the same problem of why aren't all humans the same amount of inbred?
Well, it's not a problem because when you
ask the royal family.
As the royal family. So you'd have Adam and Eve who would immediately start having kids, and then their kids would start having kids, and so on and so forth. Adam and Eve would have had dozens and dozens of kids. Now, what we see in terms of the biblical bottlenecks, we can actually reflect them on phylogenetic trees, is we see that's why you see this starburst pattern, right? Where you have explosive growth from a center point. We can see this in the mitochondrial DNA and the Y chromosome. Okay, Y chromosome is inherited from our father, mitochondrial DNA from our mother. So the point I'm trying to make is the biblical bottlenecks, they were short. They were 1 or 2 generations max, and they were followed by rapid and exponential population growth. So what that actually means by definition when it comes to bottlenecks is very limited genetic diversity would be lost. And since at creation, you'd have no genetic mutations to come to the forefront, you wouldn't have any genetic damage. And at the flood, you would have, 1 generation followed by rapid and exponential population growth. You wouldn't have the type of mutation accumulation that we see today. But the problem is, Jackson, I'd be curious as to how you answer this. Actually, I should set it up first by asking you, since today all humans are 99.99% similar, right? That means humans have low genetic diversity. This was contradictory to what the evolutionary community expected, right? Because if you're evolving from the Australopithecines up to Homo habilis, up to Erectus, and supposedly Erectus lived on the globe for millions of years, and then eventually Homo sapiens, well, every generation you have new mutations pouring into the next generation. That would be adding diversity. So we today should have high diversity, but how can we have low diversity, would you say, Jackson?
Well, I know some African populations have an incredible amount of diversity. I know that people outside of Africa have a lower amount, and I think that fits with the model of starting in Africa and going elsewhere. But what was I going to say?
But remember,
diversity. Yeah.
Well, remember, when you look at Africans and non-Africans, right? Africans and non-Africans are still human. So overall, whether you look at an African or a non-African, there's still low genetic diversity. I mean, and this goes for the uniparentally inherited DNA compartments as well, like the mitochondrial DNA, Y chromosome. For example, in the mitochondrial DNA, the average amount of differences between any two people is about 20 to 25. Now, the most is about 120, maybe 125. That would be between like me and an African. So yes, Africans have more genetic diversity than non-Africans, but overall, humans in general, both Africans and non-Africans, still have low genetic diversity, especially compared to other species. So my question is, why isn't there a greater amount of genetic diversity if deep time evolution is true?
Well, there was a genetic bottleneck 50 to 100,000 years ago of where the human population got down to a few thousand individuals, they think, based on genetics. And there are animals with lower genetic diversity than we have, and you would think it would be uniform across all species because of the arc. But like cheetahs, they got down to just a few individuals, and they're having all kinds of problems now. Same with Tasmanian devils. I would ask, why aren't all animals suffering severe inbreeding consequences if they all went through a bottleneck at the arc?
Right. Those are good questions. Your first answer was correct. So let me address that. Before I do, I just wanted to point out the source, and then I'm going to stop screen sharing for now. Because in the debate, it was brought up by, I think, Sam Jenkins, that we found human footprints in layers that are millions of years old, that we probably shouldn't find them. Here's 2 examples, the Laetoli footprints, which I'm sure you're aware of, and then more recently, footprints in Crete that were dated to 5.7 million years old. And those that discovered and have examined these footprints, they've admitted that, okay, if these were found today, let's say they were found on a beach today, they would be indistinguishable from human, as in the only conclusion you'd make is they're human. But because they're being found in millions of years worth of strata, then it's this circular reasoning going on. So now they're saying, well, we know humans were not around back then, so therefore they can't be human, and therefore what are they, right? And
yeah, if these are human, this blows our whole thing apart, right? That's
right.
Kind of
exactly. So they can't conclude, well, these are human footprints, even though that's exactly what they like. Here's an example. Here's the Laetoli footprints, right? inline big toe.
Yeah.
Here's a chip, here's a human foot. I mean, a modern human footprint perfectly overlaps the Laetoli footprint. Now they've done studies, and I've read the studies where they try. Yeah, go ahead.
It also overlaps with afarensis, the afarensis foot. They found enough foot bones to basically say that the toe was in line with the other toes. As for the Crete footprints, they're not perfectly human. They're shaped a little bit differently. They're a little bit broader, a little bit shorter.
Actually, I've seen people with some inhuman feet before. I don't know.
Well, here's the thing about that, is when it comes to afarensis, what they actually have in terms of what they think is a foot bone, or an example of what their feet look like, is, okay, it's circular reasoning, is this footprint, okay, that they've just arbitrarily concluded belongs to afarensis, and small little fragments, which many paleoanthropologists have admitted those fragments don't even belong to afarensis. They're just a human. The reason why they look human, the fragments, is because they are human. And this is the paper I wanted to show you earlier. Donald Johansen, he points out, and he is the discoverer of Lucy. He pointed out that his collection suggested that Homo and Australopithecus coexisted as early as 3 million years ago. Now, granted, this Homo could be Homo erectus, but as I've demonstrated, Homo erectus is still fully human, still considered a human, just a degenerated human. Now, they've tried to say that the stride on the Laetoli footprint is slightly divergent from humans today, which is just ridiculous. I mean, there's different strides today. If I'm striding through sand and I've got a limp or my knee's sore or something, I mean, there's so many variations in strides that to conclude that, well, this isn't human, this is, a transitional form of human based on their stride or something like that, to me is rather weak. And Jackson, before you comment on that, because I feel free to do so, your question about the genetic diversity is correct. The evolutionists will say, well, we expected higher genetic diversity. We found that humans have low diversity. Therefore, there must have been some type of near extinction event roughly 70 to maybe 200,000 years ago, right? There's debate over it. And what would have happened was humans at that time would have been downsized or reduced to between 2 and 10,000. So what happens when you have a population of 2 to 10,000 for many, many, many years, maybe even thousands of years, while you have inbreeding, which then reduces levels of genetic diversity? But my problem with that whole model is, this goes back to what we were talking about earlier, Jackson. Okay. We as creationists, we explain the origin of genetic variation as being designed and created, right? But you guys, you explain the origin of genetic variation as being the result of mutations. So this hypothetical population bottleneck, it doesn't work, Jackson, because you have a population of 2 to 10,000 humans that are inbreeding for who knows how many years, thousands of years maybe. You'd have so many negative, deleterious, and disease-causing mutations coming to the forefront that so-called hypothetical bottleneck actually would have driven humans to extinction, rather than a spreading out in all parts of the world where this so-called human population sees dominion over the planet, eventually inventing the space shuttle. This inbreeding event would have been detrimental. I don't think it's realistic. Yeah, go ahead, Jackson. I said a lot there.
Well, biologists think the minimum population size for a viable population is 100 pairs. So 1,000 wouldn't have been the end of the world, but it would have reduced genetic diversity by a lot.
But remember, you've got 2 to 10,000, not for one generation, not for 2 generations, but you'd need hundreds, maybe thousands of generations in order to reduce all of that genetic diversity in the first place. Remember that the human population before the bottleneck would have had high diversity. So now the evolutionary community invokes the bottleneck. So it reduces the human population at that time to say between 2 and 10,000. Now they require, and they never give a number because they don't know, but let's say thousands of years worth of inbreeding in order to reduce the levels of diversity. I mean, that population would have been so genetically damaged. It's not just 1 or 2 generations. That's the problem. We see an example of this today, Jackson, as you pointed out, with the cheetahs. Cheetahs are experiencing a genetic bottleneck. They're down to 7,000. Their sperm is degenerate. They're highly degenerate. Conservationists are worried about them. And we're obviously not going to expect this population of 7,000 cheetahs to suddenly spread out into all parts of the world, seizing dominion over the planet, right? They're highly degenerate. And that's an example of what that population of humans would have been like. Yeah, go ahead.
Well, the population, they think, was at one point down to a few individuals, and that's the problem. It's not that it's 7,000 today. It's that it was even lower in the past through probably natural processes, and they just barely survived.
What would have brought the cheetahs down to 3 individuals?
A few individuals. It could have been disease, could have been the ice age, and their habitat nearly got wiped out. No one knows.
It's more so hypothetical, but you're asking about the genetic diversity levels in all animals. Why don't they reflect a flood bottleneck? The thing is, they do.
Yeah.
Because we have, I'm not sure if you're familiar with the newer Rockefeller study on genetic boundaries and the CO1 gene, the barcoding gene. It's in the mitochondria. It's a highly conserved mitochondrial gene called the CO1 gene. It's highly conserved, and there's low genetic diversity in all species. You've probably heard the headline over and over again, Jackson, or maybe anybody on the panel. There's been many articles written on it where it talks about how over 90% of today's species arose or originated at exactly the same time. That's based on this genetic boundary study. So yeah, that makes sense to us because we had a flood bottleneck 4,500 years ago, where you have all these archetypes coming out, spreading into all parts of the world. So the diversity levels we see in these highly conserved genes within animal species, they do reflect the flood bottleneck. Yeah, go ahead, Jackson.
Well, I know the paper you're talking about. I don't remember all the details of it, but I remember that it got misinterpreted by one news outlet, and people ran with that. I don't remember all the details.
Well, they said it was misinterpreted because what they did is they took the genetic data and they calibrated it with their assumed deep time evolutionary history, right? So they said, well, the young Earth creationists are misrepresenting this data because, the authors of this paper are saying that the origin of these species goes back 200,000 years ago. But the thing is, when you actually read the paper, they're not basing that off the genetic data. They're basing that off of circular reasoning and calibration. When we only go by the genetic data, we can safely say that this bottleneck, where 90% of today's species arose at the same time, was actually indeed 4,500 years ago. But you have to read through the lines. You have to read through the evolutionary-based assumptions in the paper to come to that conclusion. You have to realize, okay, the date they're getting of 200,000 years, that's not based on genetics. That's actually based on the geological column. So to me, that's not misrepresenting the paper. That's just reading the paper in an unbiased way, where we're not just assuming the conclusions and assuming the evolutionary assumptions. We're looking at the empirical data, and then we're making our conclusions. Go ahead, Jackson.
Well, I haven't read the paper, so I can't really argue against it. So
no, that's fine. I just wanted to answer your question as to why we don't see genetic diversity levels representing or being reflective of a flood bottleneck 4,500 years ago. And my point is we do because we actually see low diversity in humans, which is what we would expect. God created 2 people, Adam and Eve, 6,000 years ago. That would reduce genetic diversity right off the bat. We know God created populations of animals, so we'd expect a greater diversity within animal populations. That's exactly what we see today. And because of the flood bottleneck 4,500 years ago, we would expect a lot of these highly conserved genes to be reflective of that. And that's also what we find is low diversity in animal populations within the mitochondrial DNA barcoding gene. So, I mean, to me, it's really just fascinating how well the scientific data corroborates the biblical count of origins. I haven't seen theistic evolutionists deal with it. I haven't seen old Earth creationists deal with it. And I certainly haven't seen atheists deal with it. But I've been talking a lot, so I yield.
Well, my point is the genetic diversity seems to be variable between species. Like Tuatara seems to have a good amount of genetic diversity from what I've read, or maybe not. Maybe I'm thinking of something else. But the cheetah doesn't. The Tasmanian devil doesn't. So you would think they would be all identical if they all originated from two animals on the ark.
Well, we wouldn't expect them to all be identical because you have different generation times, different reproductive rates. We have some animal kinds that were brought in 7s as compared to in 2s, depending on clean versus unclean. Larger mammals like elephants are not going to reproduce as quickly as smaller mammals like mice. But overall, the rule is we see greater diversity in animals than we see in humans. Of course, there's always going to be an exception to the rule, but the exception to the rule doesn't overturn the rule. And the example of cheetahs, of course, after the flood, you would have had a couple cheetahs, maybe. No, you wouldn't have cheetahs. You would have had the cheetah archetype, right? You would have had some cat archetype on board the ark, possibly Polarius, if you want to look that up. I think something similar to that. Which would have given rise simply through the shuffling of these front-loaded genetic differences, simply through recombination and gene conversion. You get from one cat archetype off the ark into what we see today: house cats, lions, jaguars, cheetahs. So eventually you would have had a breakaway population. That population would have changed enough where you can consider it a new species. You could say, okay, this is a cheetah, but that cheetah would have faced a different historical and environmental history than the other felids, and therefore the cheetahs might have went into a bottleneck. It's not like a one size rule fits all, right? So yeah, go ahead.
All right. Yeah, I don't really have anything to argue with. I mean, I'm going to go on to the next thing or whatever about human evolution.
Sure. Yeah. No. That's. I think that was a great back and forth. Yeah, go ahead, Dan.
I'm wondering how many specimens they have together. I was trying to look it up earlier of these different types of homos or whatever. Homo erectus, Homo habilis. I haven't seen definitive proof that they've gotten more than like basically that little pile of bones you showed earlier. I was wondering how they determined that type of diversity in the genes and that sort of thing. I was hoping you could elaborate a little bit on that earlier, but I never got around to it. I keep getting kid aggro, so I keep running off and like chasing children around.
So. I feel your pain, brother. No worries. Well, what's interesting is we don't have the genetics. I always point out this fact.
Hey, SFT.
Yeah, go ahead.
SFT, real quick. So, born 100 years late. $5 super chat says, SFT, thank you, brother. In my life, I've heard only a few speak about the truth like you do. Jackson, I hope you consider what he is saying.
Well, I
appreciate everything.
I appreciate the kind words. Born 100 years late. I find this topic of ancestry fascinating, so I've done my best to look at both sides, and I'm just not very convinced that the evolutionary side. Yeah, go ahead.
Do you want to explain this to Andrew for about the 50,000th time? He doesn't
I've got
understand.
A whole playlist of debates with Andrew just because he's still talking a big game.
He still doesn't understand heterozygosity in a front-loaded genome. He does not understand what that means. It's really amazing.
It's good practice, though, if you keep practicing doing it. And
yeah. I've only written several books on it, Andrew, and had complete debates on it. Created heterozygosity just means, in simple terms, a state of DNA diversity. Homozygosity means a state of no DNA diversity. So the original created kinds would have obviously been created in a state of DNA diversity. For example, the Bible says, be fruitful and multiply. Now, would God have wanted that to be carried out through cloning? Because if the original created kinds were created genetically homogeneous with no DNA diversity, that's exactly what would have occurred. Cloning. Okay, there's no DNA diversity built in. So for God to design organisms with the front-loaded capacity for change, for diversity, that makes more sense of God's command to be fruitful and multiply. That means the DNA differences we see for the most part between species within species, we would predict that since they're the result of initial design, they should be functional, beneficial, and useful. And these predictions have been in print for years and years and years. And as the years go on, John can speak to this. We find paper after paper of just that. DNA sequences, DNA elements that we once thought were non-functional evolutionary leftovers, they're actually showing and proving to be functional and highly beneficial to healthy life processes in the cell of all life. I mean, that's a fulfilled prediction right there. Yeah, go ahead.
Go ahead, me.
Well, I don't know. I guess
it's
anybody
a
that wants to comment.
very good point. Very good point. So real quick, so ladies and gentlemen, Patrick Alexander is the first to subscribe to the new Twitch channel. So if anybody wants to head on over to Twitch and sub to the channel, feel free to do so. Link is in the side chat.
That a boy, Patrick.
But yeah, man, this whole conversation of like, I really don't grasp why so many Evo atheists have such a hard time grasping that premise in relation to how it would, in many instances, account for what they think is proof of evolution from a phylogenetic perspective. It's like, if you have divergence and deletion of, you had their pre-existing capacity, you go down a level, and you now don't necessarily have the previous version accessible, it's going to look like a hierarchy.
Exactly.
Like, this is not that complicated. And this is what I talk about in relation to like computer software all the freaking time, and nobody seems to grasp. Although last night or the night before, whenever it was, with Deepak, who was he's a machine learning artificial intelligence developer, he understood exactly what I was talking about when I was talking about something as simple as user permissions and, you know, the hierarchies that are created in that logic in basically every software application that exists. And developers understand this. Now, some of them who are the hardcore atheists don't want to accept that the same logic applies to genetics or could apply to genetics, but it doesn't mean that it's not real. And
right, like how would you explain like apply. Yeah, like how would you explain like symbiotic relationships and things like that? I didn't mean to cut you off, dude. My bad.
No, that's a great. Actually, John, a pull up Andrew Desperate Cummings worthless argument that we've demolished a million times there, where he says recombination does not lead. Okay, so recombination does not lead to this diversity when there are only two alleles at one loci. Okay, which is a genetic location, and two individuals to start with. Can't happen. John, just out of curiosity, how many loci do you think exist in, let's say, the human genome?
Oh, no idea, but it's a lot.
A lot. Okay, so I'm going to teach Andrew here, who I've had several debates with, same with John, basic science. Okay, it goes back to this idea of just in basic genetics, big A, big B, little b. Okay, so the point is, Adam and Eve would have been created with millions of DNA positions, where what they would have is allelic diversity. They'd have capital A, lowercase a, capital B, lowercase b. Okay, you wouldn't just do this in two spots within the genome. I mean, you would have this kind of heterozygosity in millions of DNA positions that exist. Let me repeat that, Andrew. You'd have DNA heterozygosity in millions of DNA positions that exist. Okay, it's erroneous to say an allele is just two or more genetic variants when you don't actually elaborate on the fact that this is DNA variety at a specific loci. Now, because you have millions of genetic locations in the genome, you then have pretty well an unlimited potential to produce all sorts of variety, even in a single generation. Think about Adam and Eve. They got millions of spots in their genome that are heterozygous, big A, big B, little b. In one generation of recombination, where you scramble up those DNA differences, John, what are you going to get? You're going to get amazing variety, unbelievable variety. The evolutionists, they don't believe you can get variety quick, because remember, they need millions and millions of years to build up this allelic diversity in the first place, because they look to mutations, right? Well, if that allelic diversity is already built in from the start, then all it takes is 1 or 2 generations to produce variety. This goes back to my debate with Sloppy Saint a couple of weeks ago with John here on the channel. I mean, I must have tried to explain that to him 100 different ways. He just wouldn't get it because he kept trying to say, oh, no, you can't explain all the species today in a young Earth creation model. And I'm like, yeah, if you assume that the origin of all DNA varieties mutations, of course not. You need millions of years to build up the variety. But if it's built in from the start, you get visible distinctiveness in 1 generation. Does that make sense, guys?
Yeah, it makes sense to me. It's just like and if everybody else wants to weigh in, feel free. But I mean, I just don't get why this premise seems so difficult to comprehend. I really wonder if Evo atheism prevents people from actually thinking and doing an analysis of anything that actually contradicts their standard talking points. And that's just
they can't.
The more I interact with people and observe the responses and like the apparent inability to grasp concepts, like what you're talking about, it's like they think, this contradicts evolution. I will not compute, cannot compute. They go into complete like system meltdown, red spiral, or spiral death, and
like Andrew right now. Like Andrew right now. Pull up his next comment where he says, "You're not addressing what I said." Put it up. Put it up. This guy who just is incapable of addressing. Yeah, right there, right there. Okay, Andrew, again, listen. Okay, because I know most of it goes over your head. And Andrew still hasn't been able to, for one, make better predictions than the young Earth creation model. 2, he's been incapable of addressing the phylogenetic data from the mitochondrial DNA and the Y chromosome. Okay, Andrew, as I've pointed out, when we start from the genomic position definition of an allele, okay, rather than your gene unit definition, I've even pointed this out to Joshua Stramidas, and I noticed in some of his later interviews, I think he's accepted correction without admitting it, because I've noticed he's also been using the genomic position definition of an allele now as well. Okay, so let's put this into perspective. Let's take one single gene, right, John? Just one gene. Okay, well, we know that one single gene usually spans roughly thousands of nucleotides. Okay, and then you have these variants. Okay, these single nucleotide variants, you have them distributed throughout that gene. Okay, for example, let's say at 90 of the nucleotides within the gene, you have single nucleotide variants. Okay, well, that means every single one of those 90 variants, okay, that means they would have existed in a heterozygous state in each of the individuals of the pairs, let's say, brought on board the ark. And then obviously the creation event where you have more than just pairs. I mean, just from one single gene, you have a tremendous potential for allelic diversity. I mean, the question is, why don't we see even more variety today? You know,
well, the other thing,
arguments
and the other thing, something else that has to be addressed, and I know this will go right over Jackson, not you, Andrew's head, is the variability that is, we know beyond doubt, is possible from the regulatory perspective. So the caterpillar to butterfly, right? We can take on an entirely different form, not just a kind of different form, an entirely different form can happen. The genome actually doesn't change. Regulation of it does. And in fact, lots of it like melt, like literally melt and disappear. Anyway, my point is that we know beyond doubt from observed evidence that from a regulatory expression variance of gene expression through variety of regulation can dramatically change things in real time. So to somehow argue that, oh, the gene is, the only thing that could have cause a change would be, allele variant, and somehow say that's the only thing that you have to account for in terms of potential variation of species and, like body plans, all that kind of stuff, is like, to me, it's a very, very small observation frame of reference in terms of like what is actually necessary from a big picture perspective. And I think because actually looking at it from a bigger perspective, the right now, so good grief, Andrew's continuing to be dumb. Do you want to address that, SFT?
Yeah, he's misrepresenting because he's not getting it. Okay, let's take Adam and Eve, for example, okay? Just Adam and Eve at creation. So let's say they're created genetically heterozygous. Well, what does this mean? This means that they would be a mix of capital and what? And lowercase letters. Capital A, lowercase a, capital B, lowercase b. Okay, that means they were probably something like a middle brown skin. Okay. And therefore, just from that mix of capital and lowercase letters all throughout the genome at millions of DNA positions, DNA spots within the genome, they have pretty well unlimited potential to produce every shade of skin that exists on this planet today. And this is the same for the animal kinds. Okay, you bring on a cat archetype, a dog archetype, a bear archetype, you name it. All of those front-loaded DNA differences, those front-loaded functional DNA differences can transcribe and explain all the different species we see today, all the different species of bears, all the different species of felids, canids. And a prediction of this model is DNA function. And we now have over 80% of the genome. We have evidence that over 80% of the genome is active. Now, we don't know what every DNA sequence does, and that's why we have PhD young Earth creationists like Nathaniel Jensen, for example, who is making very specific predictions, saying, okay, you know, through mutagenesis, we're going to figure out that this is important to the genome, you know, this DNA sequence, therefore it's going to lead to this disease, or, you know, this gene here is not truly neutral, it's actually multifunctional, and therefore multifunctionality explains a lot of the hierarchical patterns, so on and so. I mean, you could read these papers, you can read these predictions, but these critics don't do so. It's just looking really bad for them at this point. John, did that make sense, brother?
Yeah, I mean, I totally follow what you're saying, but let's stop trying to.
Yeah. We've given enough time to Andrew, so we'll let other people jump in.
Yeah, I know.
Yeah, I was going to say he's just over here derailing. It looks like that's kind of funny.
Yeah, Slavic. You can come back in, brother. Sorry about he was waiting while we were ranting. Yeah, I don't know, Jackson. I think you'd come off mute there for a second. Did you have something you wanted to insert?
No, not really. I'm not really into genetics that deep, so I'm not the one to argue that position, really.
Well, Jackson, let me ask you a question. What would be your favorite fossil specimen in terms of what you believe is a good example of like a transitional or a basal-like human?
A human? Let me think. Probably my favorite fossil, not that it's the best ancestor, but my favorite fossil was the Littlefoot specimen, the Australopithecus prometheus, because it's the most complete.
Obviously, I have some comments to make on that, but I don't want to. Anyone else want to engage the fossil evidence with Jackson, because that's more a specialty. We'll kind of get off the genetics. I can engage him further, but I know we've been engaging quite a bit on this. So, anyone else have any thoughts or?
I actually I got a quick question. A lot of this stuff is way over my head, but I always wondered about how in the world did they know how far away we are from the genetics of like the super early humans? I mean, I'm assuming it's because they compare us to apes, just because, I mean, nobody has the genetic code of, you know, really old humans, whatever they're called.
Yeah, exactly. That's a great question. I was going to point this out earlier, but I've often made the point that when we're trying to answer this important question of ancestry, I usually label it as separate ancestry or universal ancestry, right? Then we need to look to genetics. We need to look to traits, because that's what's inherited in sperm and egg. That's how we're going to answer the question of ancestry. But yeah, a lot of these bones found in the dirt, a lot of these fossil specimens, we don't actually have their genetics. So oftentimes there's a lot of inference being made simply based on morphology, simply based on a collection of bone fragments. And that's why the fossil record itself involves a lot of fancy storytelling. The story is oftentimes changing. That's why when we got the Neanderthal genetics, we quickly realized, wow, I mean, we interbred with them. We literally have their genetics. They're human, but we don't have the genetics of the Australopithecines. We don't have the genetics of most of these so-called examples of human evolution. So yeah, that's a good question. It's a lot of inference. Also looking to the chimpanzee genome, comparing chimpanzee genetics with human genetics, comparing human genetics with gorilla genetics, so on and so forth. But yeah, they don't have the genetics of most of these fossil specimens.
So would it be fair to say it's like completely guessing at, well, because we have similar genetics code to, let's say, an animal, well, then therefore we are related. It's literally just guessing because you have an assumption that we came from them. If we didn't have that assumption that we came from them, then you can just be like, like the Ford car. I heard this, I don't know where I heard this, but like they have different models. Does not mean that they're all evolving to a newer and better model. It's just that different, one designer designed a bunch of them. So if they don't hold to that dogmatic view that we're all evolving, well, at least, different we were apes earlier and now we're a little bit more non-apes. If they didn't hold to that view, the genetic information wouldn't be pointing to that. Is that correct?
Exactly. I like the way Kent puts it, since it was Kent versus Jackson today. He says, just because the lug nuts of a Pontiac are interchangeable with that of a Chevy doesn't mean that they evolved from a skateboard millions of years ago, right? It demonstrates a common designer, just to put it simply. Jackson, I saw that you unmuted yourself. Go ahead.
Oh, I was just going to say it's getting pretty late over here. Well, not over here, but just for me. So I'm probably going to head out. If that's all right with you guys.
Of course. Yeah, Jackson, you've been busy tonight. You had a 2-hour debate and now a 2-hour after show. You're a champ, so I appreciate it. If you want, you can have some final thoughts, final words before you get out of here. But it's up to you.
No, I just thought it went good. I mean, I enjoyed it, win or lose. I haven't decided if I won or lost yet. I'll have to watch it back. I'm probably going to do that here in a minute and go to bed.
Yeah, it's pretty late here as well. I got to go make sure everybody's still in their bed too. Dude, you did a great job, Jackson. I didn't mean to steal your. I'm gonna get off too.
No, thanks. All right, well, see you guys later.
Hey, blessings, Jackson. Great debate. Dano as well. I was gonna say before he left, I liked his profile picture there. I thought that was funny. So Jackson, yeah, I'll shoot you an email sometime. And again, great debate, tons of fun.
Okay.
I appreciate it.
Yeah, it was fun. All right, I'll see you guys later.
Appreciate it, Jackson. Have a good one, man. I just stepped away for a second. Appreciate you joining the after show.
So just to kind of double check here. So most of this genetic, the stuff they talk about, or geneticists, at least the ones that are talking about evolution, they're literally making up stories because they have an assumption. It's not like it's hard science, even though they talk about it like it's hard science. Am I understanding this correctly?
Absolutely. Yeah, they don't. You'll be fascinated by this paper that I've been invoking in almost every debate for 3 years. Nobody ever addresses it. John, maybe share my screen just real quick. The genetic data where they build their stories doesn't even help them at all. Because for one, they're finding out that when we actually consider the entirety of the chimpanzee and human genome, we're really only about 88% similar genetically. And that translates into like 400 million DNA differences. Okay, so what we have is a huge number of fixation events. Fixed in genetics just means to get stuck in place. Okay, so we need a huge multitude of fixation events since the split about 6 million years ago between humans and chimpanzees. We know fixation for the most part is relatively slow. So what there is something called a waiting time problem to even get enough fixation events since the split between humans and chimps for human evolution to even be true. But more so recently, when you can see the paper here, chimpanzee, this is a secular paper, this isn't Young Earth Creation. Chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content. So here's what's interesting, is the Y chromosome is non-recombining DNA, okay? We only inherit that DNA compartment from our father. With the nuclear DNA, there's recombination, right? So it recombines, it scrambles, a little more messy. So the Y chromosome should be relatively more stable. So when they sequenced the chimpanzee Y chromosome and human Y chromosome and compared it, they were shocked. They were shocked that, notice here, more than 30% of the chimp Y chromosome lacks an alignable counterpart on the human Y chromosome and vice versa. Page's team found that chimp Y chromosome has only 2/3 as many distinct genes or gene families as the human Y chromosome, and only 47% as many protein coding elements as humans. So point is, the Y chromosome between humans and chimpanzees is only about 35% the same. It's hugely different, massively divergent. Too many differences to explain in just 6 million years since the split. Because remember, you got to consider every single human Y chromosome on the planet is nearly identical. That's how we can build these Y chromosome family trees, and we can trace our last Y chromosomal ancestor back to just 4,500 years ago, because there's low variation. But then the Y chromosome between humans and chimps is only about 35% the same. I mean, how are they going to explain that? Point is, that's exactly what we would expect from a creationist. But it's the genetics that is actually contradictory to the ape to man evolutionary story. So even the genetics is not on their side. The genetics themselves, the genetic data is refuting them. So if they go to the fossil record, okay, they're using fossils and bones to build a story that's not consistent with the genetics. The genetics comes first. That's the direct way to answer this question of ancestry. So they want to overturn the genetic data with their bones found in the dirt, their bone fragments. You see how circular and you see how unscientific that is?
Yeah, that I didn't know this part, but this is just beautiful. I mean, It's like a nail in the coffin.
Oh, it is. It is. I mean, I've debated PhDs on this, and I've brought it up to them, and their best explanation is chimpanzees have had a wild sex life from generation to generation, where they have multiple partners, and therefore, the sperm in their genetics on the male side is doing some crazy things enough to explain the divergence. But this is all just a guessing game, all a hypothesis. They even admit this is a hypothesis. Put it this way. Okay, they say that our closest cousin is the chimpanzee, okay? It turns out that our Y chromosome as a human is more similar to the gorilla Y chromosome than it is to the chimpanzee Y chromosome. Well, that ruins their hierarchy. It should be humans and chimpanzees that are closer. Why in the world do we have the gorilla closer to us? And they'll say, well, it's the chimpanzees, the odd man out, because of their interesting polygamous way of mating all throughout history. Just stories, of course, to explain the data. But I mean, there's another nail in the coffin, right?
Yeah, but so if I understood you correctly, the Y does not really recombine correctly. Is that correct?
That's right. Yeah, it's what's called by genetics uniparentally inherited DNA compartment. Just means that it's only inherited for example, we got our Y chromosome from our father. We got our mitochondrial DNA from our mother. So there's two non-recombining DNA compartments.
Okay, yeah. So technically, it doesn't matter how many partners they would have. It wouldn't really change the Y, and so their theory wouldn't really hold water.
It may degenerate it a little bit more. Yeah, I mean, there wouldn't be any recombination as a factor here. But they could also say, so we know that the Y chromosome actually mutates really fast. So here's a technical paper from Dr. Nathaniel Jensen. He's a Harvard graduate. He works at Answers in Genesis. And he looked at high quality mutation rate studies on the Y chromosome, and he realized that the Y chromosome mutates really fast, way more fast, like about 1 per generation. So this is the title of the paper. He demonstrated that the Y chromosome mutation rate and the number of DNA differences in the Y chromosome suggests a 4,500 year history, which is perfect because that brings us back to Noah, our last Y chromosomal ancestor. So the evolutionists will say, well, okay, the Y chromosome mutates fast. So that explains why the chimpanzee and human Y chromosome is so different. When in fact, it actually makes the problem worse for them. Because think about this. The Y chromosome mutates really fast, right? But when we actually examine Y chromosome diversity worldwide, we've realized that the Y chromosome, no matter what population you look at, it's the same. I mean, there's very low variation, very low differences in the human Y chromosome. So how in the world do you have a very quickly mutating Y chromosome that's also nearly identical worldwide? It should be different. There should be divergent people groups. There should be more diversity. Well, you want to know why? It's because it's young. It only goes back 4,500 years. So although it's fast, there's only been 4,500 years of a fast mutation rate. Does that make sense? It has to be young if it's fast and there's low variation. Because if it went back a couple 100,000 years, well, that would be a couple 100,000 years where we have mutations accumulating in the Y chromosome, which means the Y chromosome should have high diversity. So that actually doesn't help them either. That actually makes the problem worse. So like you said, it's nail after nail after nail in the coffin for the evolutionists.
Yeah, so it's interesting because even if that was the case, it would not just have to, like, a certain line of chimps have to be changed. They would all would have to have a similar, like pretty much one person's completely changed or one, animal changed, and everybody followed that. Everybody else died out, right? Because if it wasn't, it would still, they'd be closer to us than,
Oh yeah,
35%.
Oh yeah, it should be way. I mean, our Y chromosome is only like half the size of the chimpanzee Y chromosome. The architecture, gene content, totally different. For example, Call Me Emo in the chat says what I just said. He's an evolutionist. So here's his rescue device. He says, actually, it's the chimpanzee's Y chromosome that is significantly different. He says mainly due to losses. And that's what I said earlier, right? Is that's their story. It's the chimpanzees, the odd man out. And everybody else's Y chromosome is normal. When no, that's contradictory. The human Y chromosome mutates really fast. So if what Call Me Emo is saying is true, okay, that it's only the chimpanzee Y chromosome that's doing some wild things enough to make it so different, then we should find highly divergent people groups on this planet where the Y chromosomes are quite divergent. Okay, but no, we find one singular Y chromosome worldwide. I think there's about maybe 300 at most. It would be the max differences between any two people. So if you have a mutation rate in the Y chromosome that's 1 per generation, you can easily explain the maximum diversity in the Y chromosome in just 4,500 years. If what Call Me Emo is saying is true, we should find much more divergence in human Y chromosomes, and we don't find that. So every one of their stories and rescue devices is actually refuted by the empirical data. Like we can make a prediction. Like we can look to their rescue device and say, okay, that'll lead to a prediction. Let's see if it's true. And we find out it's false every single time.
Yeah, and another thing that you guys were talking about was the specifics of, like speciation, let's say it that way. How you can have one animal that, breaks down into, like, let's say there was a pre-cheetah, and now that is broke down into different cats. And you can technically explain it by the genetic code degenerating quickly. Yet we don't see that in humans. It's literally staying pretty similar if you look at it in that sense.
Right. Yeah. It's very similar. Right here. All human beings are 99.9% similar, identical in their genetic makeup. I mean, isn't that fascinating? Apparently, we've been evolving for millions of years, and yet we're 99.9% the same. And I was pointing out to Jackson, I was trying to find, I've got thousands of slides, so sometimes it's hard to find the exact paper, but here we go. Inbred Neanderthals left humans a genetic burden. Remember, he was talking about how, Neanderthals are slightly different than us in their DNA. Well, of course, they were the most inbred people group on the planet. Their rungs of homozygosity are amazing. and the systems and the environments that they lived in. They lived in this cold glacial type of ecosystem where the environment changed their genetics. The environment actually changed their morphology. That's why they're built the way they are, because they're adapted to that cold environment. So of course we predict them to be slightly different, but overall they're 99.7% the same to us. Meanwhile, chimpanzees are 88% the same. Call me emo in the chat. Look at this. He's going all the way to rats. So his rescue device now is this. Look, he says, Standing, do I have to bring up those rats again? So he's pointing out that there's rat species within the same genus that have massive differences, right? Well, when you look to rats and mice, just have a look at their reproductive rates, have a look at their generation times. I mean, they can recover populations quickly. Kind of like rabbits, you know, they'll say breed like rabbits, right? But we're talking about humans. And he wants to, on the phylogenetic tree, he wants to go all the way down to rats and show that the Y chromosome is doing some pretty crazy things there. He's missing the point. Yeah, I admit the Y chromosome is mutating really fast. But you have to answer the question, then why is the Y chromosome so similar in every human male on the planet? If it's as wild as they say, and it mutates as fast as we know, then it should be, I think this makes sense on the panel, right? If we're accumulating that many mutations, then if we really do go back a couple 100,000 years, it shouldn't be as similar as it is. But no, it's very much similar because the Y chromosome's only a few thousand years old. So that's really
that makes perfect sense.